Well, that depends on what kind of what the carbon is attached to. Okay? If the carbon has all hydrogens, what would we predict about how much energy it would have? A lot, the maximum amount, right? When you start replacing the carbon with oxygen, or if you replace it and convert it to a carboxylic group, there'll be much less energy. So it depends on what the carbon, the functional groups that are bound to the carbon. So fat, a saturated fatty acid of fat, has got 9 kilocalories per gram. A carbohydrate has 4 kilocalories per gram because it doesn't have all those CH2s. Okay, chapter 10. We just got over this, so I think it's fresh in your mind. Is it fresh in your mind, or is your mind gone? Gone off somewhere. So what do you need to know about the photosystem? That's really the most important thing. I think the photosystem, how they work. So the photosystem, here is the photosystem. Complete photosystem. Photosystem 2, photosystem 1. And these are the light reactions that take place. So a certain kind of light, wavelength light, hits this photosystem 2. All those pre-colored pigments, all the chlorophyll A's and B's, they gather the photon energy and direct it to what? P6A, that's in photosystem 2. P6A gets excited, goes up in excited state, electrons are ejected, drop down to ground state. Then these electrons move merrily through. They come to plastic phenols. That's like photons on a cube. Then it goes through the cytophones, which has got iron. Then it goes through copper. And as it goes from here to here, it releases enough energy to make 2 ATPs, 2 photophosphorylation. Simultaneously, water is broken down to oxygen and proton. Okay? Then, photosystem 1 gets activated by a different wavelength of light. It's got chlorophyll A's and B's and pigments too. They're all light gathering. They gather the photon energy directly to P700. It gets excited. Goes into the excited state, ejects the electron. The electron passes through a series of iron sulfur proteins, and finally the electrons go into NAD and NADPH. So in the presence of light, you make 3 compounds. Oxygen, it goes into the atmosphere through the stomata. You make ATP through photophosphorylation, and you make NADPH. Right? That's the linear electron transfer. Then, there's something called circular. That only means you make ATP. That's less important. Then there's a discussion about, on page 196, about the difference between mitochondria and chloroplast. I would sort of read that. And just sort of be familiar. It's completely analogous. Works exactly the same way. And that's demonstrated in figure 10.18. Okay, so here is photosystem 2, this photosystem 1. They're both being activated by light simultaneously, and that causes what? Protons to be coming off here, protons to come off here. You make NADPH, and the protons then move through simple diffusion. They arrive at the ATP synthase, which looks just like the mitochondrial ATPase, passes through to make ATP. So the kinetomotic theory is functioning here too. Then you have the Calvin cycle. Calvin cycle. There are only 3 compounds there you really need to know. What are those 3 compounds? The 3 compounds that you should know about is the first compound. What's the name of the enzyme which fixes CO2? Rubisco. Rubisco, the most abundant enzyme on Earth, the most abundant protein on Earth, constitutes about 20% of the chloroplast protein. That's a number to remember. So, the CO2 comes in, Rubisco goes to work, fixes it, and the first compound you make is 3-phosphoglycerate. You just saw that in glycolysis. Same compound. 3 carbons. This is, and because it's got 3 carbons, this pathway is called the C3 pathway. Okay? Then, what's the next thing? You use an ATP. You put a phosphate as a kinase, of course, then. You put a phosphate into position 1. You make 1,3 bisphosphoglycerol. Then comes NADPH. What is NADPH? It's an electron source, a strong reducing agent. And what it's going to do is, it's going to reduce, what it's going to reduce? Carbon number 1 from a carboxylic group to an aldehyde group. That's why you need electrons, to reduce the carboxylic group to an aldehyde group. So you make a bunch of glyceraldehyde phosphates. A couple of them are pulled off, and they undergo gluconeogenesis. They make glucose. And then the glucose is converted to starch, and the starch is stored in the plant, and the plant is growing, right? And you chop down the plant and eat it. Right? And you eat the starch, and you break it down to CO2 and water. It goes on and on forever. Okay, well, see, this is a C3 plant. The problem with a C3 plant is photorespiration. Photorespiration. So, this oxygen builds up inside the cell. It binds to the active site of rubisco. And instead of making a 3-carbon compound, you make a 2-carbon compound. I'm not even going to tell you what it is. It makes a 2-carbon compound, and the net result of that is you waste ATP, and this is bad for the plant. Photorespiration can waste about 50% of what's going on. Then there are plants called C4 plants and plant plants. These are plants that are adapted to cold conditions. No water, too hot. They have 2 cells, and I think you should go through that figure, which is whatever it is. Let's see if I can find the number. And if you know that figure, which is figure 10.21, I think that explains things very good. Okay, so there we are. 4 chapters. Any questions? We have 5 minutes. We're running a class next. Any questions? You have plenty of time to study for this exam. You have to start now. No screwing around. Yes.
Is the format of the test going to be similar to last time?
Sorry?
Is the format of the test going to be similar to last
The
time?
format is exactly like the first one. There'll be those unpleasant